Ernie, and this is my video for the teaching assistant position. Welcome back everybody to our second lab. This is our microscopy and cell structure lab, as you should know from the title of your lab manual. So we have a couple of things that we're going to cover today in lab. The first being how to use a microscope and learn the different parts and the functions of the microscope. We're going to use biological specimens on two different types of microscopes, a compound and a dissecting, and compare the similarities and differences. And then lastly, we're going to learn how to do a wet mount. So has anyone ever used a microscope here before? A few of you may have, a few of you may not, and that's okay. I didn't learn how to use a microscope until I went to college. So a few hands, very good. With that being said, where have you used a microscope? Where can you think of a microscope would be used for? So our top choices usually are medical research, we have plant biology or botany, we have forensics, which is a very popular, genetics is another one that falls under biology, chemistry as well, it's not listed, and also educational purposes. So microscopes have been around for a very long time. Galileo created the first microscope in the 1600s, and then Robert Hooke came along in the 1670s, changed it up a little bit, and he was able to actually look at blood platelets, which is really cool. And since then, it's advanced quite a bit. Now we have two major types of microscopes. There are more. Electron microscopes and light microscopes. Electron microscopes are a way to overcome issues of the light microscope. Light microscope is exactly what you think. It uses visible light to magnify small structures that may have, and it has a few components that we will talk about here in just a minute. Electron microscopes, there's two different types. A transition, which creates a 2D image like this, and it uses electrons instead of light to magnify an image. And the scanning it creates a 3D image, and this is an example of that as well. So we're going to go into a little bit more depth about our lighting microscope, so that's what we're going to be using today. So it has three major components that I talked about. Our magnification factor is the first one, and this is where it takes the size of our object that we can see with our own eyes, and it compares it to the size of the object under magnification. And it uses a magnifying lens to do that, and this is where we get our 10x or our 40x, and it can go up depending on your microscope. There's also our resolution. The resolution is the ability to separate objects from one another. So we can go from this to this, so it's very clear and defined. A couple things come into play with this, and that is the proper lenses, the size of the lens, the magnification factor, and also how great the user is. And finally, our focal plane. So this is done by adjusting the stage of the microscope, which I will show you that in just a minute. So it's our ability, our focal plane's right here, to move it up and down and adjusting the light, because the light is going through such a small, thin area. Okay, so our compound microscopes. This is what you may think of when you think of a microscope. We have different parts that we're going to utilize today. First, starting with our ocular pieces. This is where we look through. Then we have our diopter adjustment, where depending on how it fits on our face, we can move those to fit our eyes. Our nose piece, which has our objective lenses attached. This is where we get our 10x or 40x, etc. Our stage or mechanical stage. This is what is able to move up and down to move it closer to our objective lenses. And this also has stage control, so you can move it back and forth, you can move it left to right. We have our condenser and our illumination, which are kind of play into each other. Our condenser allows us to let light in. Our illumination is our light source, and we can adjust the brightness with that. When we carry a microscope, we carry it with the base and the arm, like this. And as we take it from our lab bench or back to the cabinet, we want to make sure we're holding it close to our bodies. It always has a light switch, and it has our coarse and our fine adjustment. Our coarse adjustment moves the stage, and our fine adjustment is really going to give us that clear resolution of the picture. And I think that's it. So a tip that we're going to learn today when we do our first exercise with the letter E, it's in our lab manual, we're going to put it on our stage with these clips right here. Just you can spread them apart or squeeze this right here to open them. Put the slide down. We're going to start with our lowest magnification, and you're going to work your way up. Once you are able to clearly see the letter E, we're going to leave our stage alone. And then we're going to move our objective lenses and then only use our fine adjustment to really make the picture clearer. And then next we have our dissecting scope. Little bit different, some parts are still there. This is for larger things you might want to look at under the microscope. So we have our eyepiece and diopter adjustment, which is the same. We have a stereo head, our objective lenses. This is different on each one. Sometimes you're able to do that on the side, or it may not rotate at all. Our light source can come from the top and the bottom with a light control, and our focus knob, and that's it. Or stage clips to hold the specimen in place, or whatever it might be that we're looking at. And then lastly, we're going to create a wet mount. So a wet mount is when we have a blank slide, and we put a drop of a specimen on the slide like this, and then we're going to take a cover slip, and we're going to hold it at one end, and then we're going to drop it down with keeping contact on this side. And then we're going to take some sort of paper towel or a chemoid, put it close to the side, and then let the water or the liquid diffuse through. So our specimens that we're going to look at today, we have pond water, sand, oscillataria, which is a cyanobacteria or a photosynthetic organism. We have a stained onion skin. This is oscillataria, by the way. And we are going to work with our cheek cells, which will be fun. And then also yogurt. And if there is time at the end of lab that you would still like more practice, there are other slides listed in the back of your lab manual. Just let me know and we can try to get those for you so you can really utilize your lab time today. And I think with that, that is it. At this time, I'm going to have you split up into groups of 2 to 3 at your lab benches, and you will need 1 at each scope. The specimens are going to be up here at the front of the bench, and if you need help with anything, please let me know. That's what I'm here for. All right, let's go.